Okay, okay, okay. So first, thank you very much to the organizers for inviting us to the conference, and for because we are a very young company. The company was just created this last spring, 2017. This is our first time here at the ITPN meeting, but this is not my first meeting here. I was here a few years ago, and I am very happy to see that the people and the conference is still very active in the field, and the mature of the projects are even better than back then. So that's really nice to see. Okay, I'm going to explain you today a bit about our company, what we are doing, and precisely what we are, what we want to do in the nanomedicine field. Okay, so basically, this is a company that it was. Oh, there's some changes between Mac and PC, but sorry for that, for the design. So basically, the company is a company that we develop drugs, early stage drug development company. This is not a nanomedicine company only. It's an early stage drug development company that basically we design novel medicines that target senescent cells. Why we target damaged cells, I will explain you in another slide. But basically, we have 3 main programs. All of them are in discovery or preclinical. One of them is with small molecules. Another one is with an antibody. And the third one is in nanomedicine, and that's the one that I would like to focus my talk today. The company was, as I was saying, I was created in Barcelona in about April 2017. Then the company was doing a due diligence with a US investor, and a US branch of the company was created in June. And finally, after a lot of painful weeks, the company was invested in a Series A summer, about August 2017. So now we have secured our funding to develop these programs and to reach clinical trials with one of these programs in the next 3 years. And that's what the mission of the company is about. As many of these companies, this is a spin-out, startup, spin-off company that comes from different Spanish academic institutions. And the 3 co-founders of the company are Manuel Serrano, who was a leading professor at the CENIO, el Centro Nacional de Investigaciones Oncológicas in Madrid. And he did a move from Madrid to Barcelona, different than the football players that normally they went from Barcelona to Madrid. So he decided to do it in the best year maybe to do it, from Madrid to Barcelona. And now he's at IRB there. And he's the person that has been developed all the biology around the senescent cells and the clinical impact of that. Together with him, the other 2 co-founders are the ones that they are providing all the chemistry and all the nano know-how around these molecules that we are designing. This is Professor Murguia and Professor Ramon Martinez, who is here with us. They are the other 2 academic co-founders behind this company. I am also one of the co-founders and the CEO of the company. And together we have been building a team that is the most important asset that we have in the company. Our team that is the ones that we are developing. We have the mission to develop this technology and reach the patient as soon as possible. Also we have building a scientific advisory board and also a clinical advisory board for each one of the 2 indications that we are working in the company, and I will explain you later. So what is the science behind? I am also learning about senescence still, because I am not an expert on senescence, but what I learned so far is that senescence is a, senescence, it's a type of cells that are happening and emerging when there's a repair process in the cellular, after a cellular damage. So it is good to have senescent cells in our body at the beginning. And this process is I don't know, it's not working the pound. Okay, so you have on a regular repair of cell damage, you have the emerge of these senescent cells. and then is the key step before recruitment of inflammatory cells and clearance and regeneration of the cellular damage. And this is something that is happening quite nicely in our lifetime. But what happens that with persistent damage, in a pathological context or with aging, this repair pathway, it becomes maladaptive. And then we start to accumulate senescent cells. So that accumulated senescent cells has been proved in vivo that is not good. It's not good for different types of pathologies, okay? And that's all the science, and I'm sorry that you cannot see that properly, but this is all the papers that has been published from our academic co-founders about proving that in vitro and in vivo that the accumulation of senescent cells leads to fibrosis diseases and other type of diseases, and also that removing that senescent cells from our body, it could be beneficial in order to regenerate the damaged cells that has been happening in our body. So basically, our therapeutic approach is following that concept. So basically, what we are planning to do is that generate drugs that target that cells and eliminate that cells from our body. That's our therapeutic approach in the company. So we design drugs to precisely target senescent cells, eliminate senescent cells from our body, and prove on a clinical trial that this is beneficial for the patient. So there's a paper that shows that this has been proved in different types of pathologies. So basically the most known is fibrosis, in pulmonary fibrosis, for example, but also in renal diseases, in cancer, in cataract diseases, in liver fibrosis, in obesity. So basically you can try to target senescent cells in different types of pathologies. And what we do in the company is that we have building that pipeline where we have two different programs focused on fibrotic diseases, specifically in radiation-induced lung fibrosis. So basically stage 3 and 4 lung cancer patients that has been treated with radiotherapy. After the treatment, that we know that 20-30% of these patients, they develop pulmonary fibrosis with time, and sadly most of them, they die not because of the cancer but because of the fibrosis. So we think this is a really interesting target in order to prove our drug. So we are developing a small molecule program and an antibody program to target this type of patients. And the third one, and it is not a coincidence that this is our most advanced program is with the nanoparticles for cancer, but this is our third program. And maybe you were wondering why is our third program, this is the most advanced program. And well, I know that you here at the conference you are all believers of the nanomedicine, but outside here, there's many concerns still about the regulation and about the cost of the manufacturing, about not in your knowledge, but also in the investors and in VCs and in pharmaceutical companies, that they are still having the perception that I feel that they have is quite risky. So I don't know if you were there, but I was in 2010. It was the first workshop, nanomedicine workshop at the medical agency in Canary Wharf. I don't know, maybe some of you were there. I was there, and I still having the feeling that from the regulatory point of view, we still are very close to 2010. So that's something that, that perception, it's becoming outside of this room and outside the nanomedicine wall, it's very intimidating for if you want to develop a nanomedicine, and that's why our investors suggest that this should be our third program, even is the most advanced one. Okay, so I'm saying that because I think it's important to be said. Okay, taking that into account, this is our program, and basically what we are trying to do is we have these fantastic nanoparticles, nanostructure that are silica nanoparticles that we encapsulate with any small molecule, or in this case rhodamine, that, and then we coat that silica nanoparticles with a galactose oligomer. Why? Because we know that senescent cells are overexpressed in the lysosomes with an enzyme called galactosidase. And the level of galactosidase in senescent cells is higher than in normal cells, and the academics designed this structure in order to be degraded only when it goes into senescent cells and not in normal cells. So basically we are building these scaffolds, and then we are able to encapsulate in this small molecules or peptides, and then we can coat that with these oligosaccharides, and then we can test it in vitro first, and that is what we did. We test normal cells, and we test with rhodamine, in that case in human fibroblasts, and then we did the same with senescent cells. So as you can see here, there's a release of the rhodamine preferentially in senescent cells. So that led us to test it in vivo. And the model that we choose first to do that, our first proof of concept, it was in a classic pulmonary fibrosis model. Bleomycin, as a cancer drug, it is the model in vivo to induce pulmonary fibrosis. So basically what we did is we inject a group of animals with bleomycin, and we induce the pulmonary fibrosis, and then we inject our nanoparticles with the rhodamine, and then as you can see here, the levels of rhodamine in the lungs of the mice that has been treated with bleomycin is much higher than the controls. Okay, so here it was our evidence that senescent cells are releasing the cargo of these nanoparticles when they are being accumulated in our body. So then the next experiments that we did is let's then prove if we can see any effect. And then what we did is, as you can see here, please take attention to this graph here. This is lung dysfunction. That is how you measure the fibrosis. So in black, you have here the control group of animals. Then in gray, we induce the fibrosis, and then you increase the lung dysfunction. So if you inject to this group of animals a drug, in this case a cytotoxic drug, doxorubicin, by itself, you are still having lung dysfunction. But if you do that with our nanoparticles, then you target the senescent cells, you are recovering lung function by killing senescent cells, by eliminating senescent cells from the body. And that was our proof of concept in vivo in fibrosis. Then the next step that we did is that let's prove it in cancer. So in that case, we use two different approaches, the silica nanoparticles with the doxorubicin, with a generic drug, and also with a BCL2 inhibitor, that is a drug that is been known to be a good senolytic drug. So again, we induce senescence in tumor mice, and then we inject our senolytic nanoparticles. Okay, and as you can see here. You see here in human melanoma xenograph, after inducing senescence with a chemotherapeutic drug, in that case palbociclib, is a CDK4, CDK6 inhibitor developed by Pfizer. You can see how we increase a lot the level of senescence by this biomarker that is precisely the galactosidase. And so then you can see our in vivos. I will explain you the doxorubicin, but it's the same case with the navitoclax, with the BCL2 inhibitor. So basically here you have a control, human melanoma xenograft, the tumor grow, and then if you inject our delivery system, our nanoparticles with the doxorubicin, but you are not inducing senescent cells, as you can see in gray, the tumor grows. Okay. Then, if you treat the tumor with palbociclib, is a cancer drug, obviously you reduce tumor growth. If you treat the tumor with palbociclib and then with our nanoparticles, then you have tumor regression. So you increase the therapeutic outcome of your treatment by combining a chemotherapeutic drug plus a senolytic drug in a nanoparticle form. Okay. So that it was our proof of concept in vivo. And right now with this project, what we are doing is we are on lead optimization. Our milestone is to launch a preclinical proper regulatory track at the beginning of 2018. And we are applying to nanomedicine translational programs in the US, because we have a company and we have an office in Boston and also here in Europe. So our idea is to partner with many academic groups, with institutions, with companies here at your conference and in another conference in Europe, in order to reduce our clinical translational risk with these nanoparticles. Thank you very much. Yes.
Thank you very much. Very promising. I have a quick question. Why the US? Where are the reasons to choose to move to the US?
Money.
Only money, or you thought?
Our investor is from the US.
Yeah, because there is a lot. You can find investors in Spain. Or the reason why you didn't receive positive answers in Spain, or?
Sorry, I didn't elaborate properly. So the idea is that this is still a very early stage company. Okay, we are not even on a proper preclinical regulatory track. So obviously the company was created in Spain in March, and different investors, local investors in Spain. I am in the board of other biotechs, so I know the ecosystem, and also in Europe they were interested in our company. But to be honest, it was more a seed funding that will allow us to do some steps. Okay. And again, the feedback about the nanomedicine, it was, okay, this is really promising, but as investors, we think it's too risky. Okay. In the other side, when we have the opportunity to do a due diligence with a US investor, we will be able to secure our investment for 3 years, and not only a seed capital, but I cannot comment on the quantity of the Series A, is confidential, but we were able to reach even a phase 1 clinical trial.
And just to ask if you feel that in US things will be more faster than in Europe, for approval, for preclinical assay, for clinical assay or so on. So did you have this feeling that you have the investor in the US, but you have this feeling that in US you will be faster, or you are thinking to do it in parallel in Europe or in Spain and in US, or you will focus effort in present things in US?
I would rather, to be honest, I would rather to do it in Europe. Okay. And it's been a few years that I have not been in the nanomedicine context, but I've been in many. From 2007 and 2012, I was involved in many nanotechnology things, and for me it will be much better to do it in Europe. But you have to take into account that 75% of the biotech market is in the US. Okay, and obviously if you get your drug approval by the FDA, you have access to pretty much the market. Okay, so things go quicker, or let's say more fast track probably in the US because of that. That's my opinion. I'm not saying that here we are slower, because I'm saying that maybe because the market is really focused on in the US, that's what's happening. Yes.
I'm sorry if you already mentioned, but you are thinking in systemic administration or inhalation?
For the cancer, is a systemic. For fibrosis, pulmonary fibrosis, maybe it will be in inhalation.
Fibrosis. Yeah.
Yes.
Thank you very much for the presentation. I interpreted your results slightly differently in the way that I assumed that that the macrophages that are part of the inflammatory milieu that you create either by the bleomycin or by the cancer, and that you, by a nanoparticle approach, deplete the macrophages rather than the senescent cells. So how critically dependent is the approach on killing the senescent cells locally, or is it just macrophage depletion?
Oof. I think I cannot answer properly to that question, but what we are seeing is that and that's more a feeling that something that I can support with data. Now we are replicating these results with a CRO, but basically and with GMP-like material. But basically, what we see is that when we tested in vitro, okay, and cell-based assays, we didn't see any specific effect that maybe you treat with an navitoclax is the gold standard senolytic drug. Okay, so it's been approved for all, is on clinical trials for other reasons and has not been going on because it has a side effect. Okay, but everyone in the senolytic works with navitoclax, and you compare your senolytic drug with navitoclax. So if you compare the nanoparticles with navitoclax or navitoclax with navitoclax inside the nanoparticles, okay, you don't see in vitro many effects, many different effects. But when you translate that to in vivo, then you see you see that the nanoparticles are doing quite effect, especially because, as I was saying, navitoclax didn't went through, it's an ABI molecule, a BCL2 inhibitor, didn't went through phase 1 because it has a side effect called thrombocytopenia. Okay, so we will be able to demonstrate, well, our academic founders will be able to demonstrate that if you encapsulate navitoclax with our nanoparticles, then we avoid that side effect. That's something that obviously the delivery system is improving, not only the efficacy, but also, as you know much better than me, reducing the side effects as a good carrier. Okay. Okay. Thank you.